Some patients respond to a T-cell therapy, then relapse months later. So we ask a question that sounds simple and isn't. Which genes actually steer a T cell into the state we want, and which ones just change the most when we perturb them? Telling those apart is the whole game, and most screens can't. A real controller answers three questions. How much did the cell move? That's reach. Which state program moved? That's precision. And did independent donors agree? That's repeatability. Magnitude alone is necessary, but nowhere near sufficient. So we silence one gene in a primary human T cell, read the whole transcriptome, and compare where the cell went across guides, donors, and contexts. The key question: does direction add anything after magnitude is already known? It does. In the pre-specified test, adding direction and repeatability to a model that already knows magnitude improves recovery of known regulators by 0.357. And because 13 positives is a small set, we also ran a separate, stricter test, a fully refit, leakage-free out-of-fold estimate, which lands lower at 0.215. These are two different measurements, not one number shrinking, and we report both, because hiding the more conservative one would be the easy lie. Here's the part I'm proudest of. We went looking for where it breaks. On a broad external set of regulators, it fails, minus 0.28, the whole interval below zero. Remove the four master transcription factors and the gain collapses, so the honest claim is narrow. Canonical state-defining regulators in T cells. And the boundary is graded. Immune T cells pass. A myeloid line is a near miss. Non-immune cells fail. That's a scope map. It tells you exactly where the property lives, which is why we never call it an invariant. A controller is not a drug target. Our top controller, IRF1, points the wrong way for intervention. And as a CAR-T response predictor, the approach is a null. A simple CD8 fraction does better. We report that plainly. So the real innovation is judgment. Every failure, every null came from a correction loop where Claude challenged our own headline, found a leak in the controls, and forced the stricter test. The code owns the numbers. The skepticism is the science. And it points to a concrete next experiment, a pre-declared donor-resolved panel that can falsify the next claim. Everything is open source. Clone it, run one command, 21 of 21 gates pass. What's left isn't a target list, it's a tested scope map with every pass, failure, and null written down. Because a good scientific agent doesn't just find an answer, it knows when not to call pass.